About a cut like this, when you cut your finger, millions of cells are generated to heal that cut, like new blood cells. And in fact, every one of the 37 trillion cells in our body came from, no matter how specialized, a single cell at conception. And the key to all of this is stem cells. Stem cells are the foundation of developmental biology, repair biology, and also excellent candidates for cell therapy. So what is a stem cell? And is there only one kind of stem cell? Well, the answer to the first question is that stem cells have two key properties. They're a class of cells that can exhibit, first, long-term self-renewal. They can make more stem cells. Or they can go down a different path and differentiate into multiple other cell types that are more specialized in their function. For example, those stem cells could become blood cells or muscle cells or nerve cells under specific cues. But is there only one type of stem cell? The answer to this question is definitely no. In fact, this term is used broadly, so when you hear the term stem cell therapy, that is really not descriptive of where did that cell come from and what might it be used for in terms of a therapeutic outcome. So to really understand stem cells and how they might be used for cell therapy, we have to take a deeper dive. So when stem cells replicate, they divide into, for example, two daughter cells from a single stem cell, and these daughter cells contain the same DNA as the parent stem cell. As a consequence, when these daughter stem cells differentiate into a more specialized cell function, say a nerve cell or an epithelial cell, they contain the same DNA as that stem cell in the parent generation and in that daughter generation, but they have very different functions, and that speaks to the different ways that the DNA is processed to lead to, for example, protein expression, generally in response to environmental cues. So the DNA is maintained, but the function and the expression of that DNA is modified by the environment, inside the body or in the lab. For example, hematopoietic stem cells can differentiate into the different cell types of your blood. White blood cells, red blood cells, and dendritic cells all come from our hematopoietic stem cells. Now, while it was once thought that stem cells were only found in embryos, which they are, and in bone marrow, which is a ready supply of hematopoietic stem cells as well as mesenchymal stem cells, we know now that stem cells are found in almost every human organ. These stem cells that are found in the adult organs are responsible for replacing the cells that are lost due to normal wear and tear, as well as responding to and replacing cells in the course of injury or disease. Now, we also know that along that differentiation pathway from a stem cell to a completely specialized cell, there are intermediate or progenitor states. And these progenitor cell states are also important and relevant to cell therapy because these states can have those cells act as drug factories inside the body, influencing the behavior of cells around them without necessarily differentiating into tissue cell types. Now, the potential of stem cells to differentiate into these different cell types is known as their potency. And it's important for us to recognize that stem cell potency is different from therapeutic potency, which would mean a correlation with a therapeutic outcome. It can be related, but really in stem cell biology, this means the ability to adopt a different phenotype or differentiate. Now, ideally, we would harvest these stem cells from each tissue type, and then that would serve as the basis or the reservoir for any cell therapy addressed to that particular tissue or that particular disease in that region of the body. But practically, this is not always possible. We can't obtain stem cells from every part of the adult body. The easiest way to recognize this is to think about the source of neuronal stem cells. So the cells in your central nervous system or your brain do include stem cells, but we can't actually obtain them. And so the next best option would be to have a higher degree of potency in cells that we could coax to act as neuronal cells, even though for many practical reasons we can't obtain neuronal stem cells from the brain. So let's talk about the different types of stem cell potency now. Totipotent stem cells are those first few cells that are formed when the zygote is created. So at this point of fertilization, you have totipotent stem cells that can differentiate into every cell type in the body and the placenta. So you can generate a whole new organism in principle from these totipotent stem cells. They have all the power, or totipotency. Next, there are pluripotent stem cells that are found in the blastocyst, the next stage of development. So they have multiple potencies, the ability to become multiple cell types, but they don't have the ability to form the placenta, so slightly more restricted. And then multipotent stem cells can still form multiple cell types, but a fewer number. And these are more specialized in terms of that developmental perspective into the different germ layers in the body, the ectoderm, the endoderm, and the mesoderm. As one example, hematopoietic stem cells can differentiate into any of the different fully differentiated blood cell types, but hematopoietic stem cells cannot differentiate into liver cells, for example. Finally, unipotent stem cells are also found in the adult body, and they exhibit long-term self-renewal. They can make more of these unipotent stem cells, but they can only differentiate into one cell type. Now, these are often the source of tissue regeneration that was mentioned earlier as a natural part of the body's repair system, and also as a potential source for cell therapy when they can be obtained. Similar pathways exist for cells that originate from the ectoderm, the mesoderm, and the endoderm lines. Now, as we progress through this differentiation decision path, the degree of potency of the stem cells decreases. The cells get more and more specialized. As a result, their potency gets reduced in terms of the pathways that can be taken. Now, while certainly not the only source of stem cells for cell therapy, one source of pluripotent stem cells is from human embryos. These are known as embryonic stem cells, and they're actually derived from the inner cell mass of a blastocyst. So this source of cell therapy would come from an actual embryo. And as a result, there are certain ethical considerations as well as strict legal requirements regarding their use, including for cell therapies. So you can see that in this context, there was a driving force to consider other ways of obtaining cells that retained certain great features of early stage multipotent or pluripotent stem cells, and this led to the development of induced pluripotent stem cells, or iPSCs. So this concept of reversing the specialized function of an adult somatic or tissue cell and retaining this capacity for self-renewal and later specialization is a remarkable discovery that was first demonstrated in 2006. In groundbreaking research in the laboratory of Shinya Yamanaka in Japan, it was shown that adult stem cells could be reprogrammed in such a way that they de-differentiate into an embryonic stem cell-like state. It's important to recognize that these are not embryonic stem cells, and there are many challenges scientifically as well as practically in the use of iPSCs as stem cell-based therapies. But there's certainly a lot of promise, and it's fair to say that this has revolutionized the field and the thinking about cell-based therapies and the sources of those stem cells. In fact, this advance was so well recognized that Yamanaka received the Nobel Prize in Medicine only 6 years after that first demonstration, and he shared this honor with Sir John Gordon, who demonstrated 40 years earlier the concept of actually de-differentiating cells through the process of nuclear transfer. Now, before we conclude, it's important to note that stem cells behave differently in the body, in vivo, compared to in the lab, in vitro. In the body, stem cells have the potential to differentiate and self-renew many, many times. That long-term self-renewal in the context of an injury or development is a hallmark of stem cell biology in vivo. However, in the lab or in the production environment, stem cells do not maintain long-term self-renewal. After a certain number of cell divisions, this capacity is highly reduced. And so it's important that we have experimental conditions, such as the media formulation or other parameters, that are appropriate and well controlled to allow these cells to proliferate without taking on unintended attributes. So in summary, stem cells are undifferentiated and they exhibit long-term self-renewal. They have the capacity to become differentiated tissue cell types or other cell functions, such as your blood cells, but Along the way, they can adapt a progenitor state that can actually have those progenitor cells act as drug factories. So for these reasons, stem cells are considered a very exciting and potential source of cell therapies. In fact, induced pluripotent stem cells adds another tool to the toolbox for stem cell-based medicines, but there are many challenges in understanding how to work with these cells in a laboratory and manufacturing environment to create potent and efficacious medicines.